Okay, we'll move on to the next talk. Is going to be presented by Jianping, Wang, and the title of the talk is using CRISPR-Cas9 and hairy root systems to explore the gene functions during peanut nodulation. Jianping, go ahead.
Good afternoon, everyone. Can you hear me all right?
Yes.
The slides and the pointer, can you see it?
Yep.
Okay, great. I will be sharing the story about using this CRISPR-Cas9 in couple with hairy root transformation system for exploring the gene function during the peanut nodulation. So why do we care about the peanut nodulation? Basically, the nodulation in peanut and also many other legume crops is contributing the biologically fixed nitrogen for the current crops and also for the following rotating crops. So which is very important for the sustainable agriculture and also to protect the environment by reducing the nitrogen fertilizer application. And the biological nitrogen fertilizer, biologically fixed nitrogen actually in the peanut counts about like 55% of the peanut nitrogen needs, and compare with many other legume crops, the level is relatively low. So it leaves a huge potential for us to increase the nitrogen fixation in peanut. And there are two different ways if we classify of the process of the rhizobia invading into the legumes. Basically, two different ways we can identify. One is the root hair invasion, and the other one is the non-root hair invasion. And for the root hair invasion, actually it happens in most, a lot of the legume species, include the model legume species and many other legume crops you can think of, like a soybean, common bean, and many other like mung bean, cowpea, except peanut. So peanut is actually one of the one in here in this category using the non-root hair invasion. But for the root hair invasion, because model legume species and too many legume crops using this invasion process, it has been extensively studied. But for the non-root hair invasion, it's extremely understudied. So peanut is the only legume crop actually invaded by the rhizobia through this non-root hair invasion. And this invasion is considered as an ancient, this kind of bacteria-plant interaction for this infection process. And it reassembles the pathogenic bacteria invasion into the plants. And also it has much less host molecular components involved in this process. So it holds a great potential as a reference pathway to engage, to engineer the non-legume crops for symbiosis in the future. So it is critical to understand the molecular mechanisms of the non-root hair invasion for the peanut nodulation. And however, it is, as what I mentioned, extremely understudied. So we refer to the root hair invasion a lot. And for example, here, this is the root hair invasion process. Basically, the root cells release the flavonoid, and that is perceived by the bacterium. So it stimulates the bacterium to the rhizobia, basically, to produce a nod factor. And then the root hair sense the nod factor and recognize it to have this calcium spiking. That signal is transducted and to cause the root hair to be curled and then to form this infection thread. And then through this infection thread, the bacteria infected the plant cells, the root cells, and continually the signal will be transmitted into the cortical cells, which stimulate the cell division to form this nodule. Basically, in the nodule is a biological nitrogen synthesis is going on there. So as we can notice, here is a pathway for the root hair invasion. And the first step here is the receptor, the NOD factor produced by the bacteria is received by the NOD factor receptor NFR1 and NFR5. These two genes are required to receive this NOD factor signal to pass on the signal to the downstream for the nodulation to happen. So our question is, are these two NOD factor receptor genes required for the peanut symbiosis, which is using the non-root hair invasion? So to answer that question, we use the CRISPR Cas9 system coupled with the peanut hairy root transformation for the gene function validation in the peanut nodulation. It goes through 7 steps. Basically, it identifies the orthologs of those two genes in the peanut genomes, and then we design the targets of those two genes for the editing, and then we make the construct to transform into the agrobacterium rhizogene first, and then we induce the peanut hairy roots from the hypocatal and transform the construct into the hairy roots. And then the next step, we identify the putatively transform the hairy roots by the GFP signal detection, and then next is checking the nodulation formation in the hairy roots to characterize whether the function for the nodulation is normal or not. And then, of course, we have to verify whether the DNA sequence editing is successful or not in the transforming the hairy roots. And we do the amplicon sequencing of the targets through Sanger method. So of those two genes in the peanut, we identified totally three homologous pairs. One is NFR5 with each copy on the subgenomes, and we identify 2 pairs of the NFR1 genes. And these genes, because of we know the subgenome, the close relationship, those 2 genes, the sequences is highly identical. We can see here. We also evaluated these 6 genes, their expression level, and we found out this pair actually the expression level is very, very low. So basically, specifically during the when the root is infected by the rhizobia, so the expression is low. So we excluded this for the CRISPR-Cas9 experiment. We just focus on these 2 pairs, and then we design the targets of the NFR1. When we design the targets, we designed 2 targets here, and both of the targets were trying to lock out both of the homologous copies because the sequence identity is so high. So we have both copies knocked out in one of the experiments for the NFR1 and also for the NFR5. We also designed 2 targets to knock out both homologous copies. So for the binary vector construction, we have the targets, which is 19 to 20 nucleotides bound, and then we threw the overlapping PCR to have that linked with the metacarpal truncatula, the U6 promoter with the scaffold. So this one is, as an insert fragment, we put the target or the single guide RNA into the vector, which is the P201G Cas9. And since we have 2 targets, so basically we have one here and the other one side by side to each other in the vector. And then we have this construct after validating, we transform it into the agrobacterium rhizogene string K599 through the heat shock transformation. And then we screen those transformants in the medium with the kanamycin for the single colony. And then after the single colony is validated, then we transform the plants. And for the plant transformation, we have the peanut seeds germinated 6 to 7 days. And then at the catalate, hypocatal region, we make the cut, have the bacteria rhizogene, agrobacterial rhizogene inoculated, which is carry the the construct, and then we grow them in the vermiculite. And then after about 2 weeks, we can see the hairy roots are induced. And then we observe those under the fluorescent microscope. If they're fluorescent, basically it tells us the transformation is successful, carry the vector. And after another 2 to 3 weeks of growing, we see the hairy roots grow more and more, and then we inoculate with the rhizobia to see whether the hairy roots produce a nodule or not to validate the function. That is the basic process, and here is the results. For the first one, we did the NFR1. Again, we have the both targets, okay, and both targets hit both of the homologous copies to have those knocked out. And fortunately, we got 12 transgenic hairy roots, which showing the GFP positive. Then we amplify the target gene for the validation of the genome editing. And interestingly, we noticed 5 of the events actually have 2 amplicon, with one which is smaller size, the other are a bigger size. So we cloned those amplicon into the vector and then pick up 20 clones randomly, and then for Sanger sequencing to see the genome editing. And here is some of the results of those small fragments, and we found out actually they are all have a big junction between the target 1, okay, which is here, and to the target 2 up to here. So all of the regions have been basically deleted or removed or cut. The size is about 341 or 42 nucleotides, and which is similar for the other events of these 5 small fragments. And of course, we also sequenced the longer fragments, the P1, P up to P3 are the events which we transform just the empty vector. And this is the wild type, and here is the water control. So for the other events, okay, we also sequence the target, and here is different events we can see have different types of the editing, and some of them are have different type of editing, and like for example, these have added as a target 1, and the target 1 has the deletions about 9 nucleotide, and here is 1 nucleotide deletion, and here will be 28 deletion, and then the target 2 have 1 deletion, and 3 clones shows this type of mutation, and 7 clones shows this type of mutation. This is how you read the data here. And then the interesting thing is every single event we can see different versions or variety of different editing scenarios actually is mixed together for every single event. You can see here. And particularly I want to point out is these 2. Every single event we have the genome edited on either the target 1 or the target 2, but for this one you can see there is no editing, 2 columns here on both targets, and these 2 are edited, which means of these hairy roots, okay, this event, some of the cells, some of the fragments are edited, some of them are not. So with this, we summarize all of the results, and here is what we get. 10 out of the 12 events were successfully edited, and mosaicism, like with a mixture of different type of the editing, actually was observed in 9 out of 10. Majority of them have this kind of mosaicism, and 8, which includes these completely edited ones, still produce a nodule, which basically tells us this gene is not required for the nodulation in peanut with the non-root hair infection.
Okay, just under a minute to go.
Story of the second gene here. Similarly, we got 10 transgenic hairy roots, And here is the editing results. And the conclusion here is 8 out of the 10 events were successfully edited. And similarly, we have the mosaicism was observed in all of the 8 events. But the interesting thing is 1, 2, 3, 4 different events was completely edited, and they all cannot produce nodules. And with this result, we conclude the NFR5 actually is required for the nodulation. So here is the conclusion. This system is an effective method for studying the gene function involved in nodulation. However, the mosaicism of the hairy roots actually challenge the efficiency of these methods, and we have to generate large amounts of this transgenic hair roots to be able to conclude. And the NFR1 is not required, but NFR5 is required for the peanut nodulation, which is the difference between the root hair invasion and the non-root hair invasion. And with that, I would like to thank Dr. Guo and Dr. Juman for providing the seeds, and this work is mainly conducted by